Excellent.
Phenomenal. So can everybody see everything?
Looks good.
Okay, so are we ready? Should I begin presenting?
Please take center stage.
Okay, so my name is William Hofstedt. My mentor was Dr. Patrick Dagnan and his PhD student, Jericho Ortenius. I will be presenting on the fluorescent tracking of horizontal gene transfer in gut microbes. So the human gut microbiome is pretty diverse. Here's the entire abstract of our project, just in case someone watching the recording wants to read the whole thing, but some highlights to make this more time reasonable is that changes in the microbiome of the human gut can contribute to both health and disease. Horizontal gene transfer is largely responsible for these changes, such as what nutrients these microbes can consume and how they avoid an immune system. Tracking horizontal gene transfer, though, can be tricky, and in this experiment, I will be proposing an examination of these events using fluorescence. So really, the goal is to identify MGEs by using fluorescence-based alternatives to antibiotic gene reporters. So in the past, methodologies of identifying MGEs were slow and narrow. They were largely homology searches or guess and check methods with lots of sequencing involved and were ultimately very costly. A solution or an alternative methodology is to use transposon mutagenesis to find larger numbers of MGEs all at once. So the way this happens is that you insert genes essentially into random locations into a population. So every cell in that population has the gene inserted into a slightly different location. And this is done using PSAM suicide plasmids. Then you can mate that group with a second group that is genetically uniform, and by examining the genome of the second group to see what DNA came with the reporter, you can essentially get an idea of where to start looking for mobile genetic elements in the original group. And as I said before, the methodology we are using relies on fluorescence instead of antibiotic reporters. Good reasonings for this usually involve that a lot of strains we work with are already resistant, and we need to overcome that resistance and find their NGEs regardless of that resistance. And a second good reason is to avoid creating antibiotic-resistant bacteria in the future, just in case there's a lab leak or they become pathogenic, various reasons like that. So our general hypothesis is that if we use fluorescent gene reporters and place them into many random locations in the genome of a donor gene pool, then they can be used to observe the movement of mobile genetic elements and help us isolate recipient cells. Now, we've made a lot of progress to date. This project is actually almost done. We've just stalled in the last couple of steps, and we're currently working on overcoming those issues. So to start in our project, we have tested a few fluorescent proteins to use for our reporters. Most of them were ineffective, but two of them were effective. There was UNAG and IFP. UNAG was eventually used for our donor population, and IFP was used as to mark our recipient population. And the true transconjugates that are resulted from mating the donor and recipient populations could be found by checking what cells show both IFP and UNAG. IFP was already shown to be effective, so most of my testing revolved around UNAG and testing its effectiveness. So UNAG and UNAG side by side with the other tested proteins. I kept them on two separate graphs, mostly because they were incomparably different. If you look at the y-axis of each of these graphs, you can see the one on the right is going up by less than 5. The one on the left is going up by thousands. Fluorescence is a measure of intensity in this case. So it's just relative intensities is what we're focused on here. I have already generated the donor and recipient populations. Of course, the donor population is now a transposon mutant library with a wide variety of different mutants with the insertion in a random variety of locations within the cells. and the recipient population is genetically uniform, with the end goal being a mating. Now, the mating has been unsuccessful so far. There doesn't seem to be any significant gene transfer between our donors and recipients. Now, this has changed a little recently, but that was literally last week, so it's not in this presentation, which I started making a month ago. To overcome this kind of roadblock we've encountered where our two species aren't mating, the obvious next steps are to try using different donors or different recipients. It's possible that these two just do not mate very well. They don't transfer their mobile genetic elements to each other. They might need closer relatives. The current ones we use are both Bacteroides species. So there's Bacteroides theriamycin and Bacteroides ovatus. There are closer relatives that we could try using. We could also allow more time for cell growth because we are testing the efficacy of our fluorescent protein methodology by double checking it with antibiotics, which are used in parallel. So we've essentially connected the antibiotic resistance to the fluorescence so that we can check the fluorescent methodology using the antibiotic methodology at the same time, with the eventual goal of being able to eliminate the antibiotics entirely. So assuming we can overcome this roadblock, some expected results are to be able to completely verify that the fluorescent methodology matches the results of the antibiotic methodology. And assuming that is successful, flow cycler analysis can yield the ability to find successfully transformed recipient cells within a solution of unsuccessfully transformed recipient cells. So flow cyclers, of course, they essentially dilute cells until there's only one cell in each drop of fluid, and then each drop of fluid is dripped through a laser. And that laser essentially records what colors that drop is fluorescing. And those are my references of the cassettes used were created by Liza Doe, previous PhD student, and a lot of other works were referenced for creating a lot of the materials used in this experiment. And I believe that's all.